So we are having our next speaker. This speaker is dying from a different country, China. So this this event is representing a really global thing, right? So in the morning we have people dying from Poland. Now it's the 8:30 something in China. So in the morning, so we have Cheng Li, a physician scientist dedicated to translating basic research discoveries into transformative therapies. With over 20 years of research experiences and 13 years of clinical practice, he co-founded Rectigen Therapeutics in 2016 to advance his pioneering work on RNA-mediated gene activation into clinical trial. He is a former associate professor at UCSF and professor at Peking Union Medical College Hospital. So I visited his company in Suzhou, China, just last summer with Dong. We were very, very impressed by his innovative delivery and RNA targeting platforms. When I first saw his SOD1 mouse survival data, I almost couldn't believe it because it's too good to be true. It's 200% more effective compared to tofersen. So I went there with Dong in the morning. We spent 5 to 6 hours, very intensive discussion, throwing very, very difficult questions. But at the end of the day, I believe I was convinced this is such an innovative delivery system and also the new modalities. So we hear a lot about antisense. You will also hear a little bit antisense, but it's in a different way to treat ALS. So without further ado, I would like introduce Cheng to introduce his new delivery RNA therapeutic delivery platform called SCAD.
Okay, give me one moment. I'm sharing my screen. Can you see my screen? Hello? Can you hear me?
Yes, we can see your screen.
You're
okay,
ready
great,
to
great.
go, definitely.
Okay. Thank you very much. And then wait for your invitation and for your last introduction. It's my honor to join this event from let me see. It's not advancing from the beautiful city of Suzhou here. Okay, everything good. Rocketgen is a clinical stage multi-platform company founded in 2017 on the siRNA modality or technology. A few years ago, I think 4 years ago, we thought we have the technology we can deliver duplexing RNA to the CNS. Why don't we tackle ALS with our technology? So obviously, the first target we want to work on is a sort of one, and very quickly, this program advanced from a concept, from idea to IND enabling, and to patient within just less than 3 years. So of course, this success was built on the foundation of what Dong has done in this field, and also the many other work have done by in the antisense oligo field. And to build on the success of the SOD1 program, we quickly launched another program targeting fast ALS, and this program is now also in, we have reached the PCC stage, waiting for advancing this program to IND enabling. And of course, the major focus of our work still is on saRNA. So we have a saRNA program now in phase 1 trial, which is an oncology program. We have another saRNA program. The we use saRNA to turn up a gene for the treatment of DMD, and this program is now in IND enabling. As you can see, your Rockgen is really focused on a lot of rare disease. So today I'm going to introduce our SCAT delivery platform, which is enabling several CNS programs and focus more on the ROC17 program for treating ALS associated with SOD1. So oligonucleotides fall into two major categories, single-stranded ACL and duplex RNA, including siRNA and saRNA, which is what we are working on as our main technology. The key difference arises from the backbone modification. So ASOs use uniform phosphorothioate backbone or PS modification, and this promotes strong plasma binding, protein binding, leading to broad tissue distribution and in vivo activity without specialized delivery. So in contrast, Dilfersong can only have limited PS modification, their function, which restrict the bioavailability. As a result, effective delivery systems are required for therapeutic efficacy, particularly outside the liver. So currently, FDA approved the siRNA drug designed to for liver target delivery using platform like LNP and GalNAc. However, delivery RNA to extrahepatic tissues, especially the CNS, remain a significant challenge for RNA. So this situation underscores the critical need for innovative delivery platform to enable effective CNS delivery of RNA therapeutics. So we know that siRNA is generally more potent than gapmer ASL for gene knockdown, as demonstrated in this example. So the two siRNA targeting SOD1 gene were almost 100 times more potent than the gapmer ASL targeting the same gene in culture cells. And this ASL use the in this comparison match this sequence in chemical modification, not toxin. So the key challenge now lies in how we could translate this superior in vitro potency of siRNA into effective in vivo activity. To address this issue, at Rockgen, we developed the SCAT, which means Smart Chemistry Aided Delivery, and it's a simple and efficient delivery system. SCAT leverages the self-delivery property of ASLs through a unique design that links a single-stranded oligonucleotide, which we call accessory oligo or ACO, to one of the strands of the duplex RNA payload. Its modular architecture allows independent optimization of the payload and the delivery component. When administered locally to confined space like the CNS, non-SCAT achieve broad biodistribution and long-lasting activity. It's compatible with both siRNA and saRNA delivery. So for the CNS, SCAD enable dual-RNA delivery via intrathecal injection with no dosing frequency. Potentially every 3 to 4 months, once every 3 to 4 months. And this technology is undergoing clinical validation and advancing multiple CNS program at Rockgen Therapeutic. So the process of creating a SCAD-enabled drug candidate is very straightforward. You can first off your duplex sequence in vitro with chemical modification, and then conjugate one of the strand with the ACO. And these graphs show the scarification process and how it retained very well the potency of the 5 siRNAs, highlighting the benefit of modular design. And of course, it's also possible to start with fully chemically modified duplex and the stratified duplex at the design and the screen stage. To evaluate SCAD siRNA in vivo, RNA was administered in mice via ICV injection at 20 nanomole. So after 14 days, it achieved over 80% SOD1 mRNA lockdown across the CNS regions, including the spinal cord, indicating broad diffusion, while peripheral tissue like the liver show minimal activity. So in comparison, the same siRNA without the delivery component exhibited only marginal activity. So we did directly visualize the distribution of size 3 labeled as SCAD siRNA after ICV injection. 3 days after injection, we could see broad and uniform size 3 signals throughout the CNS, including the brain and the spinal cord. So we also evaluated biodistribution of SCAD siRNA in rat by in situ hybridization. 14 days after single intrathecal dose, the Anderson strand of the duplex was detected in different parts of the CNS, including spinal cord, as shown here as an example. So next, we wish to know how long SCAD siRNA tissue exposure and its lockdown activity could last. So mice were sacrificed at various time point up to 5 months after single ICV dose. In the motor cortex, which is near the injection site, the drug concentration peaked immediately. Here, as shown here, followed by a slow decline with substantial level remained at 5 months. So here, the red line represents the SCAD conjugated siRNA. The blue line is the siRNA, the same sequence without the delivery component. So you can see the duplex along also showed high and durable drug exposure in the motor cortex. So now let's look at the PD activity. So both the siRNA and the SCAD siRNA peaked at the 2 weeks, but the SCAD siRNA demonstrated significant deeper and more sustained lockdown. So despite the high drug exposure, siRNA lockdown activity rapidly returned to baseline. So this is the site near the injection. Let's look at the deep brain structure. The substantial higher drug concentration was achieved by the SCAD siRNA with peak lockdown activity at 80%, which sustained very well towards the end of the study. At the most distant part of the CNS from the injection site, drug concentration was lower overall than in the brain, but still detectable at the 5 months. So despite the lower drug exposure, peak activity was still reached over 80%, and residual activity lasted till the end of the study. So in the liver, there was an initial high drug exposure, which declined very quickly to undetectable. Similarly, there was a initial moderate PD activity, which returned to baseline in 3 months. Due to slower CSF turnover in large animals, such as monkeys and humans, so the durability observed in mice may translate to even longer lasting effect in monkeys and in patients. So indeed, in the CNS of crop eating monkey, after 2 biweekly dose of scattered siRNA, over 80% peak mRNA knockdown was seen in both high and medium dose groups. And this activity was very well maintained over the 70-day observation period. So interestingly, CSF SOD1 protein level continued to decline at the last time point, suggesting a long-lasting PD activity of scattered siRNA. So we anticipate that even longer durability could be seen in patients. So most of the data I present so far have focused on the SOD1 gene. But SCAD technology is broadly applicable to and can target any gene with a duplex sequence. So here, a SCAD siRNA targeting the tau gene was tested in PS19 mice, a model of a tau path. 2 SAV dose of 10 nanomole administered on post-natal days 179 and 254 significantly reduced phosphorylated tau protein accumulation in the hippocampus and the cortex to an extent close to similar to the wild type animal. So this demonstrates SCAD ability and the delivery siRNA to deep brain structure effectively, preventing or clearing toxic protein buildup. So next, I would like to introduce our ROC17 program powered by the SCAD platform. So ROC17 is a siRNA targeting the SOD1 gene aiming at treating ALS associated with SOD1 mutation. And this drug is administered to patients via intrathecal injection. And this program has received IND clearance from both FDA and the Chinese counterpart. IIT study has been completed with 6 patients treated, and a formal phase 1 study is currently ongoing. So it's widely accepted and the clinically validated concept that mutations in the SOD1 gene leads to toxic gain of function. So ROC17 works by degrading SOD1 mRNA, reducing the synthesis of both mutated and the wild type SOD1 protein. And this approach helps to decrease the accumulation of toxic aggregates associated with the disease. In several preclinical studies using the SOD1 G93A model, our SCAD siRNA has demonstrated remarkable therapeutic efficacy. For example, in this single dose efficacy study, the model mice were given 1 single ICV dose at PND 60 compared to the vehicle control. SCAD siRNA significantly sustained body weight and extend animal survival in a very nice dose-dependent manner. So similarly in rat, SOD1 G93 ALS model, SCAD siRNA treatment also significantly improve the muscle strength and extend the survival. Here we use a control duplex with the same SCAD architecture as a negative control. Here, the blue line, the red line is our ROC17 treated rat, which has similar performance as the white type rat, as indicated by the green line. At the end of the study, the remaining rat simply didn't die, so we determined to terminate the study because we already observed significant benefit of the treatment. As we know that the SOD1 G93 mice is one of the most used model, ALS model for studying therapeutic efficacy, not just for SOD1 ALS, but also for sporadic ALS. And this model typically shows signs of disease onset around the PND 100 days and reaches terminal stage around the PND 160. So in the literature, in order to demonstrate the efficacy, treatment is typically given before disease onset. So to our best knowledge, no one has ever tried to initiate treatment after PND 90. So encouraged by the remarkable treatment efficacy demonstrated in mice and rats study I've just shown, we decided to challenge the status quo and treat the mice way after disease onset, which is more relevant to clinical situation. So we give 2 doses of our drug, PND 126 and 151. And we also include a lot of early treatment group as a positive control. So reproducibly, early treatment demonstrate remarkable benefit in improving muscle function and extending survival, as indicated by the blue lines. As we anticipate, late treatment also shows significant efficacy in both improving muscle function and extending survival, as denoted by the purple lines. So this result give us a huge confidence that our drug could translate into clinical meaningful benefit in patients who have already developed symptoms. Building on the strong preclinical effects data, we investigator-initiated trial, or IIT, in April 2023. And this is a multiple ascending dose study with the primary endpoint being the safety, focus on safety and the ALS function rating score, and the secondary endpoint including CSF SOD1 and plasma NfL concentration. In this study, 6 patients have been treated with the longest treatment duration reaching 40 months. All the participants have completed the stage 1 dose escalation phase, with some now on stage 2 maintenance dosing. So I'm sorry today I can only present the interim result from this study. Because the final study report is planned for the 2025 AAN meeting in April. And this slide provides an overview of the dose regimen for all 6 patients at the end of the study. For the first 3 patients, the starting dose was 60 milligram per dose. So after 3 loading dose, the patient transitioned to bi-monthly maintenance dose. After seeing very favorable safety signal from the first 3 patients, The starting dose for the last 3 patients was increased to 90 milligram per dose. So after 2 loading dose, the patient transitioned to maintenance dose every 2 or 2.5 months. The highest dose reached for the first patient is 180, and for the rest of the 5 patients is 150. The interim safety data indicate that ROC17 is safe and very well tolerated. All the AE reported were mild, including muscle tremor and headache, with no DLT, no SAE observed. Notably, the AEs occurs only after the first few doses. And in later doses, patients report no AE at all. And this is very encouraging that this drug is very safe, even at when it was escalated to a very high dose. So interim biomarker data indicate that the ROC17 treatment significantly lower CSF SOD1 protein level, reaching 50% reduction after 4 doses. The ROC17 treatment also significantly lower plasma NFL levels, reaching over 60% reduction after 4 doses. So very encouragingly, ROC17 treatment appears to have stabilized or even improved ALS functional reading score compared to baseline, suggesting potential clinical benefit. Similarly, ROC17 treatment appears to have a positive impact on patients' lung function, further supporting its potential therapeutic benefits. So building on the strong preclinical and patient data, we are expanding our CNS pipeline using the SCAD platform. So a second ALS program targeting FAST mutation, as I have mentioned at the beginning, is now progressing towards IIT study in China. We also have programs targeting tau for AD and the SARA program for spinal muscular dystrophy. So in summary, the SCAD platform is a very reliable, efficient delivery system for RNA therapeutics targeting the CNS. So ROC17 has shown to be safe and well tolerated at dose up to 180 milligram, with evidence of target engagement and reduced plasma NFL level and stabilized or improved disease progression. So lastly, SCAD enabled rapid development of new RNA therapeutic for ALS and other rare CNS disease. Thank you. I'll stop here.